Scientists at 23andMe, Stanford, Harvard, and USC compare Black American DNA before the slave trade to what it looks like today. The difference is a complete biological record of 350 years of violence, more specific than any document, more honest than any history book. Every missing haplogroup is a people the plantation erased. Every European signal in the paternal line is something that should never have happened. They had the data for years. Most people never heard what it showed. Today you're going to. Drop a comment where you're watching from and hit subscribe. To understand what changed, you first have to understand what was there before. Before the transatlantic slave trade began in the early 1500s, the people of West and West Central Africa carried one of the most genetically diverse collections of DNA on the planet. This is not a contested point. Every major institution in the field, from Stanford to the Max Planck Institute, confirms the same thing. Africa is the most genetically diverse continent on Earth. The variation within African populations is greater than the variation between any two groups anywhere else in the world. The reason is simple and ancient. Modern humans evolved in Africa and lived there for the vast majority of their 300,000-year history. Every other population on Earth ascended from a relatively small group of people who left Africa roughly 60,000 to 70,000 years ago. All non-African populations are essentially genetic subsets of Africa. Africa held the original library, and everyone else left with a single chapter. West Africa in particular was a genetic crossroads. Hundreds of distinct ethnic groups, dozens of language families, thousands of years of internal trade and migration produced one of the most genetically layered regions on Earth. The Yoruba of what is now Nigeria, the Mandinka and Wolof of Senegambia, the Igbo and Isan of the Niger Delta, the Congo people of Central Africa, the Akan of Ghana, the Mende of Sierra Leone. Each group carried its own distinct haplogroup signatures, its own biological fingerprint, shaped over thousands of years of distinct history. That was the baseline. That was what existed before the ships arrived. Now here is what happened to that genome over the next 350 years. Between roughly 1500 and 1850, more than 12.5 million people were forcibly removed from the African continent and shipped across the Atlantic. Of those, approximately 360,000 arrived directly in what would become the United States, roughly 4.5% of the total. The rest went to Brazil, the Caribbean, and Spanish Latin America. Of the 12.5 million who were loaded onto ships, at least 1.5 to 2 million died on the crossing. The Middle Passage was not a journey, it was a gauntlet. And the people who survived it arrived on the other side of the ocean with something no human being should ever have to carry. The knowledge that they had been severed from everything that defined who they were. No name would be recorded, no ethnic identity preserved, no language permitted to survive formally. The historical record treated these 360,000 people arriving in North America and the millions of their descendants born into bondage after them, not as individuals with histories and lineages and genetic identities, but as property. The DNA, however, kept a different kind of record. Before we go any further, if you're new here, subscribe right now. What we're covering today is the kind of science that changes how you see history permanently. Hit subscribe and let's keep going. In 2020, a team of researchers at 23andMe, led by population geneticist Stephen Micheletti, published a landmark study in the American Journal of Human Genetics. They had compiled genetic data from more than 50,000 consenting 23andMe customers on both sides of the Atlantic and cross-referenced it against the transatlantic slave trade database, the most comprehensive historical shipping record of enslaved African voyages ever assembled. What they were trying to do was answer a specific and extraordinarily difficult question. Can we see the slave trade in the DNA? Can we look at the genome of a person of African descent living in America today, compare it to DNA from people in specific regions of Africa, and actually reconstruct what happened, who came from where, what routes they traveled, what was done to them, and who survived? The answer was yes, and what they found was, in Macheletti's own words, something he and his team could not believe. The first thing the data showed was confirmation of origins. The genetic connections between Black Americans and specific regions of Africa aligned remarkably closely with what the shipping records show about where enslaved people were taken from. Nigerian ancestry, specifically from the Yoruba, Isan, and Igbo groups, showed up as one of the strongest and most widespread signals in Black American genomes. Congolese ancestry from the Congo people of Central Africa was the second most prominent. Coastal West African ancestry from the region spanning modern Ghana, Ivory Coast, Benin, and Togo was also strongly represented. But then came the first shocking finding, the hidden in that confirmation. Nigerian ancestry was massively overrepresented in Black Americans compared to what the shipping records would predict. The historical manifest showed that relatively few ships departed directly from Nigerian ports to North America. So why did so many Black Americans carry Nigerian DNA? The researchers brought in historians to help explain it, and the answer revealed a brutal detail of the slave trade that had been only partially understood before. Enslaved people from what is now Nigeria were not shipped directly to North America in large numbers. They were first taken to the British Caribbean, to Jamaica, Barbados, and the smaller islands, where they were worked, traded, and then re-exported to the American mainland as the transatlantic trade tightened. The DNA picked up a whole shadow economy of human trafficking that the written records had only partially captured. Then came the disappearance. And this is the part that stops you cold. The Senegambia region was one of the primary sources of enslaved people during the earliest waves of the Atlantic trade. The shipping records are clear. Significant numbers of Senegambian people arrived in North America, and Senegambians were prized for a specific reason. They were expert rice cultivators. They had developed rice agriculture in West Africa over centuries, perfecting cultivation techniques for varieties of rice that European colonists did not know how to grow. Plantation owners in the coastal lowlands of South Carolina and Georgia specifically requested Senegambian enslaved people to work their rice fields because they knew how to grow the crop. The knowledge of an entire farming civilization was extracted from these people along with their labor. So Senegambian people arrived in documented numbers. They built the rice economy of the colonial American South with their hands and their expertise. And then they largely vanished from the genetic record. The Micheletti study found that genetic connections between Black Americans and the Senegambia region were far lower than the historical record predicted, dramatically lower, and the explanation was devastating. Those rice plantations in coastal South Carolina and Georgia were death zones. The low country was riddled with malaria. The rice fields were flooded and stagnant. Mortality rates on those plantations were catastrophically high, so high that enslaved people in the low country could not reproduce fast enough to maintain their own population, let alone grow it. Senegambians arrived. They worked. They built an industry. They died. And their DNA, in large measure, died with them. The genome of Black America is missing an entire population, not because they were not there, but because the conditions of their enslavement killed them before they could leave descendants. That is not history you read in a document. That is history you see in the absence of a signal in a genome, of people who were present, who contributed, who suffered, and whose biological trace was nearly erased by the very system that exploited them. But the disappearance of the Senegambian genetic signal is only half of the story. The other half is what appeared in the genome that had never been there before. Every Black American alive today carries European DNA, every single one. The question genetic scientists have been studying is not whether it is there, it is universally present, but how it got there, in what ratio, and through which line. The 23andMe study answered all three questions simultaneously. And the answer confirmed what historians had long documented and black families had long known, but that had never before been measured at this scale in biological data. The European ancestry in black American genomes overwhelmingly came through the paternal line. White men, not white women. When researchers compared the Y chromosome, the genetic marker passed strictly from father to son, to the mitochondrial DNA passed strictly from mother to child, they found a stark and asymmetric pattern. The maternal lines, the mitochondrial DNA, were predominantly African. The paternal lines, the Y chromosome, carried European haplogroups at striking rates. Research from family tree DNA found that approximately 35% of African American men carry a European Y chromosome. 35%. More than 1 in 3 black American men carry, in the most direct biological measure available, the DNA signature of a white male ancestor. That is not ancestry. That is the genetic fingerprint of rape, encoded in the human genome and passed down through generations. The Micheletti study quantified the scale of what happened. In Central and South America, where different colonial practices were at work, the ratio was even more extreme. Researchers estimated that in some regions, approximately 15 African women contributed genetic material for every 1 African man. In Brazil and Cuba, governments actively implemented policies called branqueamento, meaning racial whitening, explicitly encouraging or coercing women of African descent to have children with European men, with the stated goal of diluting African ancestry through reproduction over generations. The policy had a name. It was written down. It was official. And the DNA shows it worked exactly as its architects intended. In the United States, the mechanism was different, but the biological result was similar. Enslaved women were raped by slave owners and their associates systematically across generations. Enslaved men were forcibly paired with enslaved women by owners who wanted to expand their workforce without paying for new arrivals. When researcher Joanna Mountain of 23andMe, who co-led the study, reflected on the findings, she said that the repercussions of what happened during slavery continue today in the DNA. That line is worth sitting with. It is kind of amazing, she said, that we see it so strongly in the genetic evidence. Amazing is one word for it. Now here is where the science goes somewhere nobody expected. In 2023, researchers at USC and Stanford, led by Professor Jaslyn Mooney, a descendant of enslaved people herself, alongside Noah Rosenberg and Jonathan Pritchard at Stanford, published a study in the journal Genetics that asked a question that had never been asked before at this level of precision. How many ancestors does a typical black American have? Not in a genealogical sense, in a mathematical genetic sense. Going back to 1619, generation by generation, how many separate African people and how many separate European people on average contributed genetic material to the average black American alive today? The answer was staggering in its specificity. A randomly selected black American born between 1960 and 1965 is descended from, on average, 314 African ancestors and 51 European ancestors, stretching all the way back to 1619. 314 African people, 51 European people. These are not symbolic numbers. These are statistical estimates derived from genetic data, cross-referenced against historical records of the slave trade. And when the researchers looked at when the European ancestors appeared in those family trees, they found that they did not appear randomly throughout history. They appeared during the period of enslavement, clustered in the years when slavery was active, when sexual violence was systematic, when black women's bodies were legally defined as property and had no protection under any law in any American jurisdiction. Jaslyn Mooney described the personal weight of this work with a clarity that no scientific paper alone can carry. Her father's side of the family is African American, she said. She can trace her mother's side back centuries, to the Jewish expulsion from Spain, to communities in New Mexico in the 1600s. But her father's side hits a wall. Very quickly, she said, you can no longer trace anything because of the lack of records. The 1870 census was the first in which black Americans were listed by name. Before that, They were recorded as property, or not recorded at all. This is the brick wall that genetic science is now carefully and painstakingly beginning to dismantle. So what does the full comparison look like? What does Black American DNA tell us, side by side, about the before and the after? Before, an extraordinarily genetically diverse West and West Central African population, hundreds of distinct ethnic lineages, Wolof and Mandinka from Senegambia, Yoruba and Igbo and Isan from Nigeria, Congo from Central Africa, Akam from Ghana, each group carrying thousands of years of distinct biological history shaped by different environments, different diseases, different diets, different migration patterns. After, a population whose African ancestry was partially shuffled, not by choice, but by force. Specific groups were extinguished from the genetic record by death rates on specific plantation types. Others were overrepresented because of intracolonial trading routes that no shipping manifest fully captured, and layered over all of it, universally, without exception, a European signal written in the Y chromosome distributed across paternal lines, a biological record of coercion so consistent that it appears in every single Black American genome, regardless of how African the person appears or identifies. That is what slavery did to a genome. It didn't just separate people from their names. It didn't just destroy family records. It reached into the biological blueprint of an entire population and rewrote pieces of it, not as metaphor, literally, measurably, in base pairs that can now be read by a machine. But here's what the data also shows, and this is the part that matters just as much. It survived, despite everything. Despite the Middle Passage, despite the mortality on the rice plantations, despite the systematic destruction of African names and languages and family structures, despite centuries of legal definitions that classified human beings as livestock, the core of what was there before is still there. The West African lineage that is the foundation of Black American ancestry is ancient, and it is intact. The Yoruba maternal lineages, the Congo paternal signatures, the Igbo mitochondrial DNA passed from mother to child through generations of women who were never given the dignity of a recorded name. The 2023 Catoctin Furnace study, which identified nearly 42,000 living genetic relatives of 27 unnamed enslaved individuals who worked at a Maryland iron forge between 1774 and 1850, proved this in one of the most emotionally powerful ways genetics has ever demonstrated anything. Those 27 people were buried without full names. They were listed in no census. Their lives were treated as economically significant only insofar as their labor had value. The researchers could not even determine most of their names from existing records. They were, in the eyes of the system that owned them, nobodies, ghosts. And yet their DNA survived. It reproduced. It scattered across America over 2 centuries. It passed from parent to child, generation after generation, through Reconstruction and Jim Crow and the Great Migration, and every subsequent chapter of American history. And 200 years later, it showed up in 42,000 living people who can now, for the first time, be told, this is who your ancestors were. This is where they lived. This is the community they belonged to. Henry Louis Gates Jr., who helped lead the historical analysis alongside the genetic team, called it a step toward restoring ancestry knowledge to communities whose family histories were deliberately destroyed. The genome does not forget. The genome does not erase. The genome keeps a record that no court, no plantation ledger, no census, no law, no policy of racial whitening, no act of violence could permanently destroy. What scientists found when they compared Black American DNA before and after slavery is not a simple story of loss. It is a story of what survives, when systems designed to erase people are measured not by the documents they produced, but by the biology they could not control. The difference between the pre-slavery baseline and the post-slavery genome is real, measurable, and historically specific. Certain lineages were thinned or erased by disease and death. An entirely foreign genetic signal was inserted through violence and coercion. The distribution of African origins was scrambled on a global trafficking system that moved human beings like cargo across 3 continents over 3 centuries. And yet, underneath all of it, the oldest threads are still there. Africa is still the dominant signal in Black American DNA. The lineages that stretch back 100,000 years, that trace to the populations who gave rise to all of modern humanity, that predate Rome and Greece and Egypt and every other civilization that history has chosen to center, those lineages are still there, still intact, still readable, still real. That is what the data shows. And that is what shocked everyone who looked at it closely enough to understand what they were seeing. If this video changed how you understand something, share it, drop a comment, tell us what landed hardest. Subscribe now if you haven't already.